Let me just go ahead and click record there so don't forget to do that. Yeah, so let's talk just briefly about what's going on this week. You can see there, I have the weekly overview page up for week 3. Today is the first of 2 classes on heredity. And then your master in AMP was due right now, so hopefully you did that. Wednesday's class will be the second part of the heredity stuff, but that is the first part of the material for exam 2. Okay, so what we cover today will be the last bit of material that's on exam 1, and then Friday's class will be exam 1. Okay, so for lab this week, it is a synchronous lab. We'll be going through the axial skeleton, just like we did previously with the appendicular skeleton. We'll go through a little bit of it. I'll send you out with your partner again. You'll do some studying with your partner. Come back. We'll go through a little bit more of it. Okay, so I would expect probably the two hours, maybe a little bit less this time for lab this week at your scheduled lab time using that synchronous lab session Zoom link. Okay. Again, so for lab, make sure you have your handout with you during that lab session. Printed is best unless you have another device you can have it on, which would be fine too. And then make sure you have your textbook or any other lab resources that you have, like if you bought one of the optional lab manuals or Human Anatomy Atlas or anything like that. Yeah, so that's for lab this week. Also wanted to say on Wednesday, we don't have a scheduled synchronous meeting like we normally don't on Wednesdays, but on the Wednesdays leading up to exams like this one, I will hold an optional review session for you guys on Zoom. Okay, so it'll be 1:30 on Wednesday. I haven't added that to the Zoom link in Canvas yet, but I'll do it today, so that'll be there. Okay, so if you want to come on Wednesday during our normal class time, you can come, but it's just going to be a time for me to answer questions. Okay, so if nobody's got any questions, then we'll just be done. But I'm sure some of you will have questions, and then I'm happy to go through some of that stuff and help you out with whatever you need to as we're preparing for the exam. Okay, and yeah, and so there's a few things. I went over both of the lecture quizzes you've taken so far, the Friday lecture quizzes, and the big thing on the first one was the pH one, the pH balance one, where it said, you know, here's this buffering equation. If we add a strong acid to it or a strong base, you know, which way does that move the equation? That was kind of the toughest one that people missed a lot on that quiz, and that'll be something I'm happy to go through during that review session. And then on a quiz 2, the one that was the trickiest one that people missed more of was the stuff on osmotic pressure. So you put a cell into a solution that's hyperosmotic, hypoosmotic, which way does water flow, things like that. So this is just a little piece of, hey, I know, yeah, this is kind of the bit of a quiz review that I'm doing here, but then I'll dive into those 2 topics for sure on the Wednesday review, and then also answer any questions you have. But those were the 2 kind of ones that on each quiz that people had more trouble with. Okay, and then for Monday's class, I just wanted to, so a week from today is Labor Day, right? And some of your classes might still be having classes, but I'd say we'll just go ahead and do an asynchronous day for Labor Day, even though it's scheduled to be synchronous. Okay, so I'll just do a recording just like I would normally do for our Wednesday class on that day. Okay, so you don't need to show up on Labor Day for our normal synchronous time this week. Okay, all right, I think those were the main announcements I had for this week. So there's no lab quiz this week, right? You had your lab quiz last week, and then we'll study about the lab quiz the week after the following week. Okay, I did go ahead and fix those things that I should have fixed on IRAT last Friday. So the 2, everybody, yeah, nobody marked off those 2 were messed up on the IRAT. So your quiz was kind of a little bit, or let's get back first. Yeah, those 2 corrected. So yeah, so I think we're going to go to the study guide for exam 3. You can see it over there under nurse in the 3 module. You can see study guides there right above exam 1. Hop on there. It doesn't give you a ton of information, but it does kind of help you on the key conceptual piece for your second. And the bigger things like process of repetition, the process of synthesis, or talking about information flow and things like that. Those things will the things that are more conceptual and more like step in nature. Those are the things that would be likely to show up on the longer questions, like 5 questions, so kind of long fill in the blank or long matching type questions. There are a couple of these quiz, so you definitely expect those for the exam. And the stuff that I highlighted in the study guide are the things that tend to show up in that format on the exam too. So any questions on that? Or any questions on anything on that this week? Okay, so the Friday's class is the exam. You don't need to hop on Zoom, okay? There's no Zoom Friday, but the exam needs to be taken at 1:30 at our normal time, okay? That's when it becomes available. But you won't be able to be on Zoom and on or off at the same time because they both use your camera, right? So you're just going to be on and off, taking it, but you have to start at 1:30, and have that minute time limit. Okay, any other questions before you go on? Okay. All right, maybe we're getting the hang of this thing. No, there's a lot to a lot of moving parts in this class, and I know you have 3 or 4 other classes. So, yeah, I'll have to answer questions via email as well, or wait right now after class, and I'll hang out on Zoom for a bit and answer any questions you have too. So let's hop into this stuff on heredity. Okay. Make sure, yeah. All right. Okay, so we're going to talk about kind of the basics of heredity, and it's really this is kind of the what's called the Mendelian genetics, which is really doing the Punnett squares you've probably done before in high school biology class or something like that, and really doing dominant and recessive traits. And it's kind of a simple approach to genetics. There's a lot more that we've learned over the past many years, and we find out now that most things are transmitted or transmitted. Most things are passed on from parent to offspring in a very complex manner. But there are quite a few things that are passed on in a kind of simple manner to look at. So we're going to start off with just kind of talking about history of it. There's Austin Monk named Greg Mendel from the 1800s. This is where Mendelian genetics comes from. He kind of posed this theory of genetics, and it really kind of helps scientists figure out how certain traits are passed on from parent to offspring. Right? And a couple key factors, a couple distinguishing things within this are that each parent carries 2 distinct units called alleles. Okay? So,
are you supposed to see your screen?
Sorry, Tim. Thank you. No, thank you.
Yeah.
Thank you for saying that. Okay, thank you very much for saying that. Anyway, share, that must have got unshared. Let me click on the share. Thank you. Okay, I think we're back, and you can still see my screen, right? Good. Okay, so yeah, I definitely don't. I'm not going to read through these this terminology and these definitions for you, but these are terms and definitions that you should be familiar with. Okay, so we've talked about some of these already. The allele, just talked about that. This idea of a dominant or a recessive trait. Okay, we're going to be talking about that a lot today. Dominant trait means that only one of the alleles needs to be there to show its effect. A recessive trait means that you need both. You need 2 alleles for it to see its effect. Okay, and then this distinction between genotype and phenotype is an important one that I'll talk just briefly about. So genotype is the genetic makeup. It's when we're, like, for example, when we're looking at an X and Y trait, a trait that's carried on the sex chromosomes, it's just the X with whatever trait is there. So we're talking about the color blindness one. You had a couple questions on during the Master and AMP. So the X with a capital R and the Y without anything for that trait. That would be the genotype, okay, just kind of how we scientifically code it. But then the phenotype is actually how it shows up, right? That shows up as color blindness or not color blindness, right? So genotype is kind of the genetic kind of code, the way that we would describe it scientifically. And then the phenotype is how it actually appears, how it shows up, what the trait actually looks like in the person. Okay, homozygous is going to mean that the person has identical alleles for a certain trait. It could be either homozygous dominant, meaning that both of them are dominant, or homozygous recessive, meaning both of them are recessive. Okay, and then heterozygous is just the opposite, where we've got one dominant, one recessive for a certain trait. So we'll look at some different examples of those here in a sec. Any questions on any of those terms so far? Okay. All right, so some basics of heredity here. So the principle of segregation is one principle that's kind of important to understand here. So during fertilization, each parent passes on one of their alleles for that trait. Okay, so instead of having the 2, those 2 alleles that they have are segregated into 2 separate gametes, or 2 separate either sperm or egg cells. Really, it's going to be 4. We'll talk about that more next time. And those are segregated away from each other. So just because they're together in dad doesn't mean they stay together in the offspring. And that's the kind of the point of the principle of segregation. Okay. And then this idea of dominant recessive inheritance. Certain traits are passed via dominant alleles, like we just talked about, and only one's needed. Certain are passed via recessive, and then both alleles are needed to actually see that trait show up in the phenotype. Okay, the one that we can actually see in the way that the person looks, or the traits that the person has. Okay, so we're going to use these Punnett squares to figure out the probability of an offspring having a certain trait. When we'll see the genotype of the parents, we'll use that to figure out the probability of a certain genotype for the offspring. And hopefully this is mostly review from a biology class you had in high school or one that you've had here at Point Loma. Okay. All right, so this is a list of dominant recessive traits that are determined via dominant and recessive inheritance. Okay, a bunch of just kind of random ones, right? That are you able to roll your tongue? Like kind of all these weird ones that scientists have figured out. Oh, this is actually driven by this certain genotype, right? Double jointed thumb, widow's peak. We used to, if you were here in person, we had done a lab in the past where you would have done some of these. Like we have these little strips that have this chemical on it, a PTC taster one, and some people can taste it, some people can't. It's really just determined by these little alleles and which allele you got from your parents, right? Couple here. Huntington's disease is an example of a fatal dominant trait. Okay, and so it doesn't really, that disease doesn't really manifest itself until at least in the 20s, sometimes early to mid 20s. And so they're, most dominant fatal diseases don't usually last that long, because if the person dies, then obviously they don't typically produce an offspring. But since this one shows up a little bit later, there's still a chance of producing an offspring first. And that's why Huntington's disease still stays around somewhat. But there's all kinds of examples there. The dominant ones on the left and then the recessive ones on the right. Okay, so you don't need to memorize those in any way. It's just to kind of show. Okay. All right, so let's dive into some examples here. Okay, so here's what we'll do. This is the one I was just talking about, the Huntington's disease. And the question is, what's the probability that the child will have Huntington's disease in each? Okay, so we have two different ones. We have this one is one mix with one set of parents here, and then number two is a different one. Okay, so to have Huntington's disease, all we need is one capital H, right? So go ahead and work through this one here, and then I want you to send in the probability of an offspring having Huntington's disease in number two here. Okay, so we'll do that on your clicker app. It's going to be a numeric type question, so it's a little bit different. Okay, it'll probably be similar to how you put it in with that short answer question we did before. So go ahead and work through that one, and you're just going to fill in those squares. And then if it's 2 out of 4, that'll be 50%, right? If it's 1 out of 4, that's 25%, okay? 3 out of 4, 75%. That's how it works. So go ahead and put in the percentage in your clicker app, and you don't have to have the percentage symbol. Just put in 25, 50, 75, 100, whatever it might be.
And this is for if they're likely to get it, not if they won't get it.
This is, yeah, so with this, it's if there's a capital H in there, then they have it.
Right, okay, yeah. Cool.
And we're just doing it for the second Punnett square?
Just for the second one, yes, thank you. Go ahead and work through both of them, but send in the second one. And there'll be some of these I send you into your breakout rooms for, but there's a bunch of them, so we're not going to do that for every single one, but do that here in a sec. Okay. All right. Go ahead and send that in. I'll give you about 5 more seconds. If you haven't sent it in, please do. I see 69 of you and 64 people that sent it in, so about 5 of you haven't. And remember, if you're still having trouble figuring out how to use this app, then you've got to get that figured out, because each day you use this, you get 2 points of participation credit for that. Okay, so easy, free points, but it's a way to keep you engaged during class, answer some questions. Okay. I'm going to go ahead and show the results for that. So what'd you guys say? Good. So about, yeah, almost all of you, well, 57. So 88% of you are saying 75%. Okay. So let's take a look there. So let's start with the first one here. And so we just fill in the Punnett square like this. All right. So from here, we're just taking down the capital H from there, lowercase from there. This one, lowercase, lowercase, right? So you just fill it in like that. So in this first one, we're just counting the ones that have the capital H's, right? So there's 2 with capital H, 2 without, right? So that's going to give us a 50% probability of having an offspring with Huntington's disease. So this is a parent with Huntington's disease mating with a parent that does not have Huntington's disease, right? And then the second one around, you guys said 75%. So this is a parent with Huntington's disease with another parent with Huntington's disease. They both happen to be heterozygous, meaning that they don't have 2 capital H's, they just got 1, right? They're both heterozygous for that. Okay, so we got, this is what we end up with. So has it, and then doesn't there, but that is 3 out of 4 there. So that's going to be our 75% that most of you said. Okay, good. Any questions on that before we keep rolling? That's kind of our simplest example when we're using a dominant trait there. Good. Okay. All right. I'm going to go ahead and keep moving on here. Okay, so the next one here, this is an example with a recessive trait now. Okay, so recessive trait means we need both of them to be the lowercase one in order to actually see that phenotype expressed. Okay, and so, or to actually see the phenotype. So albinism is the trait here, and so we've got two different matches here again. So the homozygous dominant with a carrier parent, and then a carrier parent with an albino parent, okay, or a parent with albinism, okay? So same idea here, fill this in, go ahead and send in the second one again. So what's the probability that the child will have albinism in each? Good, so probably about 20 more seconds or so. Most of you have already submitted that. Okay, I think maybe 2 or 3 more of you to submit. Go ahead and submit. I'll close it in about 5 seconds. All right, gonna go ahead and close that out. Okay, let's see what you guys put here. Good, so again, 88% of you are saying that 50%, so 2 out of 4. Okay, so let's walk through this one. We'll do number 1 first. So again, we would fill it in like this. All right, so in this case, hold up your hand for what the answer is for number 1. Yeah, most of you are saying like this, 0, right? Double 0. Got it. Okay, that's it, right? So there's none of them there that just have 2 small a's, right? So no chance. If we have a carrier parent and a parent that's not a carrier and doesn't have albinism, no chance. Okay, but then in the second example with a carrier parent with a parent with albinism, we do have a chance, right? As you guys just said, and we've got 2 possible. 2 there, 2 out of 4, so that's 50%. One thing that's important to clarify here is the difference between probability and prevalence, right? So probability is new with each offspring, right? And that's the same. So every time these couples produce an offspring, they have the exact same probability of each of these scenarios, right? That doesn't mean that if they have 10 kids, 5 of them are going to have albinism and 5 of them aren't. It doesn't mean that. That would be prevalence, right? So because the probability starts over every time, prevalence is what actually happens in actuality, but the probability was just the chance of having each one each time, but it doesn't always, those 2 don't always match up perfectly. Okay, any questions on that or anything else so far as we're moving? Okay, so this next one gets a little bit more complicated, so I'm going to send you into the breakout rooms here in a second after we talk about this. But another important principle in Mendelian genetics is this principle of independent assortment, okay? And so again, this is meaning that each allele is sorted independently of all other alleles, okay? And so this kind of clarifies that. Just because an allele from one parent passed down doesn't mean that all the alleles from that parent are going to pass down, okay? They're all sorted independently. So just because you got eye color from your dad doesn't mean you're going to get hair color from your dad too, right? So that's kind of an example of that, okay? So some will come from the grandpa's side, some will come from the grandma's side. It's another way to look at it, just one step back. Okay, so we're going to start with this one, and this one is looking at eye color and vision together. Okay, so as an example where we're talking about two separate traits, but again, not everything has to come from just one side. You can get some from mom, some from dad, right? And so we're going to look at two traits that have to do with the eyes. Okay, so farsightedness is dominant. Okay, so if you have one of those capital F's, then you're going to have farsightedness. Okay, you're going to need glasses to see up close. Okay, and then brown eyes are dominant over blue eyes. Okay, so if you got any of those capital B's, you're going to have brown eyes. Only way you'll have blue eyes is if you have two of the lowercase b's. Okay, so here's what we're going to do. Let me just kind of walk you through what this is going to look like. So on this slide here, you're going to fill in both of these Punnett squares. Okay, and then you're going to fill in each of these down here. Okay, so these parts down here. Okay, and then you can answer this question: what's the likelihood of a blue-eyed kid? If you want to answer that, we're not going to answer that one on the clicker thing just yet. So I'm going to send you into the breakout rooms here in a second. Okay, and so let me just go down here. Just so I can go to the next slide without showing you all the answers on this slide. So then this one here, okay, this is how you're going to now combine the probability of each. Okay, so you're going to take that number that we got from FF and put it there, number we got from BB and put it there, right? So all those FFs there, all those BBs there, right? So you're going to do that, and you're going to multiply those together, right? And they're originally going to be out of 4, but now since when you multiply fractions, you just go numerator times numerator, denominator times denominator, denominator. Sorry, getting a little mixed up in my words there. So that's going to give you out of 16, right? So you're going to work that all the way down. You're going to do all those math problems, okay? And now we've got to translate that to phenotype, right? This is genotype, but we need to translate that to phenotype. So which of those make for farsighted kids with brown eyes? How many out of the 16? How many out of 16 are farsighted with blue eyes, okay? And then we're going to convert that in our calculators to percentages, okay? And then this is the one I finally want you to report out on the clicker app, okay? But I'm going to send you into your breakout rooms. Please help each other on this. It looks like there's about 88% of you that got the first part nailed, okay? But that means there's 12% of you that don't, okay? So please help each other. If you got questions for your group, then ask them when I send you into your breakout rooms, okay? And then and make sure everybody gets to this part too, okay? This is kind of the next step. We're multiplying fractions, okay? All right, so let me send you into your breakout rooms here, and it'll probably take just a sec. Here, I was last, I'm sorry. Sorry about that. All right.
Oh wait, I feel like I did something wrong. Yeah, I did something
Going,
wrong.
guys? Doing all right?
Yes.
Yeah.
Good. Cool. Hey, guys.
Hi,
How's it going?
good.
Good.
We're all done.
You got it all done? Cool.
Yeah.
So you guys submitted on the app there too?
Yeah.
Cool. Perfect. Yeah. You can hang out and keep chatting if you want, or you can hop back in, but I'll bring you back in a minute here.
Okay.
Cool.
Okay.
Okay, cool. Looks like everybody's back now. Yeah, doesn't look like everybody's submitted on the app though. Go ahead and submit the answer that's on the next slide here. So this one, likelihood of a blue-eyed kid with normal vision. Go ahead and submit that on the app. It's like maybe about 20 more of you still need to do that. So submit that number. And we'll just start with the previous slide, kind of walking through this. Okay, so this is going to be similar to how we did it before with the with the match here, but instead we're just doing it twice, right? So we've got Punnett square on the left here, looking like this. Okay, Punnett square on the right should look about like that. Okay, and then we just fill in those fractions down there at the bottom, right? So how many times do we have two capital F's together? 0, right? So 0 out of 4. How many times do we have a capital F and a small f? We got 2 of those and then 2 small f's. So that's just a simple way to fill those in. Okay, so the likelihood of a blue-eyed kid, if we wanted to answer this question here, what's our likelihood? Which genotype are we looking for? So this, again, is the phenotype, right? And then these over here are the genotypes. So which genotype are we looking for to have a blue-eyed kid there? Somebody yell it out.
The lowercase b.
Perfect, those 2 lowercase b's, right? So the likelihood there is going to be 1 out of 4 or 25% probability. Okay, so we got that one. So questions on those 2 so far? Okay, looks like there's still about 4 of you, maybe now 3 now that haven't sent in that final answer on the next slide. So, okay, maybe now just 1. All right. Okay, so now we're really just taking those fractions from the previous slide and plugging them in here, right? And this is all set up nicely for you. Obviously, if you had a question like this in the test, you would have to set it up that way yourself. But this is all set up for you. So really just plugging in those fractions from the previous one, right? So how many FFs did we have, right? How many BBs did we have, right? So we're just going like that, right? And then we take 0 times 1. Obviously, that's going to give us 0. And then the 4 times the 4 gives us a 16. 16 is always going to be there, right? So we're just going to work our way all the way down with this, right? Okay, so those are all of our genotypes here, right? So these are all the genotypes. Okay, and then we're going to convert that to phenotype, right? So far-sighted with brown eyes. Okay, how many of those did we have? We have 6, right? So far-sighted is dominant. So anytime, and so is brown eyes. So anytime we have a capital F and a capital B in there, that's going to be included in this first one, right? Because those are the 2 dominant traits, okay? Or 2 dominant alleles. Okay, second one is farsighted and blue eyes. So now we're looking for anything with a capital F and anytime we have the two lowercase b's together, right? So there's going to be two of those. Okay, now we're looking for normal vision and brown eyes. So now this time we're looking for anytime we see the two small f's together and anytime we see those two small f's paired with anything with a capital B, right? Because that'll give us the brown eyes. Okay, with that we've got 6 out of 16. And then lastly, normal vision. Okay, so again, the two small f's with blue eyes, which would be the two small b's. So the two homozygous recessive, and there's 2 there. Okay, so we can convert those into percentages just by using your calculator, right? So 37.5 for the 6 out of 16 and 12.5 for the 2 out of 16. Okay, so then likelihood of a blue-eyed kid with normal vision. So that's going to be blue eyes, normal. So that's this bottom one down here, right? So it should be 12.5. See what you guys said. Good, so about 3/4 of you are saying 12.5. We got a couple that are doing it this way. You guys can't see my mouse, but you just didn't convert it. So you put in 0.125, okay? For the exam, you'll want to convert that to the percentage format, okay? And a couple of you put in 0.12 as well, okay? For the exam, please just move that decimal place 2, okay? Move it 2 to the right. Just like you would if you wanted to figure out your grade in the class, right? And you know you've got 85 points out of 100. If you take 85 divided by 100, that's going to give you 0.85, but you would say you have an 85%. You have an 85.0 because you times that by 100 to put it in the percentage format. So do that same thing when you report these on the exam or on quizzes, okay? All right, questions on that one?
On an exam, would we be expected to do this without a template?
Yes. Yes. Yeah. And so on the exams, that brings up a great question, Cam. Thanks for asking that. Is that you're able to use scratch paper? Okay. So there's a feature on HonorLock. I think you probably had to, you probably had to do it with the lab quiz too. I think where you can show a piece of paper that here's my scratch paper I'm going to use. You just have to show that it's blank before you start, right? And then you can use scratch paper for that. So you'll definitely need that for this exam, drawing these Punnett squares, doing all this math, right? So you'll want to make sure that you have a piece of scratch paper available to you, in particularly for this exam, but really for any exam or any of those lab quizzes where you're using HonorLock, it's a good idea to have something just so you can jot some things down if you kind of need to work something out. Okay.
Okay. I have another question.
Go ahead, Eddie.
Instead of doing like all that calculation, can we do like the 16 square Punnett thing?
You definitely can. Yeah. Well, if you've learned another method in another class, feel free. As long as it gets you to the same answer, that's the bottom line. Yeah. I don't teach it that way in this class. Just kind of give the one this way. But yeah, there's more than one way to do it for sure.
Cool. Thank you.
Yeah, you bet. Okay. Any other questions on that? Okay. All right. Well, let's keep rolling then. Okay, so then another principle here that's important to look at is called codominance, okay? And so this is a slight diversion away from our simple dominant recessive traits that we've been looking at, okay? And so the two different alleles both show their effect, even if only one copy is present, okay? So each allele is distinctly expressed, and it's not a blending, it's not something in the middle, it's actually you see them both, okay? So the phenotype is affected by both alleles. And so let's show an example of that. Okay, so with our blood types. Okay, so we all know that you have AB blood types, right? AB positive, AB negative. Those things, we're not talking about the positive negative part yet, but we're talking about the AB specifically. Okay, so what we use for this is this I. Okay, and then on the I is either a capital A or a capital B. Okay, and so if that A shows up anywhere, it's going to make the red blood cell have an A antigen on it. Okay, if the B shows up anywhere, it's going to make the red blood cell have a B antigen on it. They don't cancel each other out. They don't create something different when they're together or when they're with another one like it. They both show up. Okay, so if somebody has this genotype, the I's with an A and a B, they're going to have both AB antigens, which would produce type AB blood. Okay, we'll talk about O in a second. Okay, so if we have a mother with AB blood mates with a father with AB blood. That's this example here. Okay. So let's walk through. Yeah, actually we got time. Why don't I send you back into your breakout rooms? Do that one together. You guys can chat about it, and then I'll bring you back in probably about 90 seconds or so. Oh yeah, and answer that question there in your clicker. Sorry.
It's a likelihood of AB type.
Okay, perfect. Okay, cool.
Go ahead and send that in on your app there. Looks like about 10 more of you need to do that still. Okay. All right, it's almost everybody. Okay, good. Everybody's back now. So yeah, it looks like 1 or 2 more of you need to send that in your app. So go ahead and do that. Let's walk through this. So Punnett square. Yeah, so we can actually didn't have the Punnett square here, but we can fill this in, right? We've got IA, IB, IB, got the 2 B's there. A, we've got the A and the B. Okay, so then that's the genotype there. We can fill in those fractions. So 1 out of 4 homozygous A, 2 out of 4 heterozygous, and then 1 out of 4 homozygous B. Okay, and then those phenotypes, we're going to have 1 out of 4 with type A blood, right? That's one that just has the 2 A's. 1 out of 4 type B blood, that's one that has the 2 B's, and then 2 out of 4 with AB blood. Okay, so the likelihood, what you guys said, so 95% of you are saying 50%. So good, getting closer there to almost everybody being on the same page. So this one here, right, is the answer here. That's going to be 50%. Okay, questions on that one? All right, well, let's keep going here. So another kind of variation of this whole system is called incomplete dominance. Okay, and incomplete dominance is when a heterozygous organism shows a phenotype that's kind of somewhere in between dominant and recessive phenotypes. Okay, it appears to be kind of a blend of the two. Okay, and it's still same kind of unit-to-unit inheritance. And we'll give an example here. Okay, so sickle cell anemia is a perfect example of that. Sickle cell anemia is somewhat common among populations of in Africa around the equator and their descendants. About 1 in 625 African Americans have sickle cell anemia. Okay, and what this is a mutation in the gene that codes for hemoglobin, the protein inside of our red blood cells that helps us hold on to oxygen. Okay, so it looks like this. Hemoglobin S is the sickle cell one. Okay, so that would be the one leading towards sickle cell anemia, the actual disease. Normal hemoglobin looks like this. It's a H with an A. Okay, and so the pathology here is when you have this HS when the blood cells get a little bit low oxygen levels, it makes the hemoglobin crystallize and turn into these long kind of rods stuck in the red blood cells. It makes the blood cells kind of bend and curve into the sickle cell, or into the sickle shape. Okay, so it's kind of like this half moon type shape. They get stuck in places, they burst, and it's a very debilitating disease to have. Okay, so that's the disease, sickle cell anemia. Okay, but then the thing in between is called sickle cell trait, and this is when somebody is heterozygous. Okay, so they've got one normal and one with the actual disease genotype. Okay, so they don't fully crystallize due to the presence of this other one, the other hemoglobin that's normal. Okay, and so they may have minor symptoms, but what we know is that this actually, having sickle cell trait, actually protects these people from malaria a bit. Okay, so we're going to do this parental cross on the next slide. Okay, so two people with sickle cell trait get together, and so we can walk this one together. So again, both of these people would have sickle cell trait because they've got one normal, one sickle cell, one normal, one sickle cell. So they both would have sickle cell trait. They wouldn't have major disease here, but they would have that protection from malaria. Okay, so let's walk through it, what it looks like. This is what our Punnett square should look like. Okay, so then people who are HAA, so it should be completely normal, we've got one of those out of 4, right? And then we've got people who have sickle cell trait, or offspring options for sickle cell trait. That's half of them, 2 out of 4. And then with sickle cell anemia, so down here, 1 out of 4. Okay, so the likelihood of sickle cell trait in this case is 50%. Okay, likelihood of sickle cell anemia, 25%. Okay, so this is that example where the heterozygous type of genotype is expressed differently than both homozygous on either side. Okay, any questions on that one?
What are the B's?
The B's is just another way to, this slide, it's just another way to do the hemoglobin one. So you don't really need that. The previous one just had the H, but some people also express it with HB for hemoglobin instead of just H. So they're not alleles. They don't have anything to do with the actual anemia or sickle cell part. It's just another way to do the H thing.
Okay, cool. Thank you.
Yeah. Any other questions on that one? Okay. Okay, so another option here is something called multiple alleles, okay? And this is when there's more than just 2 alternatives. Everything we've talked about up until now, there's only been 2 alternatives, right? The capital or the lowercase, right? So in these situations, which is really true of most traits, each parent still only has 2 alleles, but there's more than 2 possibilities, okay? So this is what happens with blood type, okay? I said we would talk about O here in a second. So with blood type, we have the A, we have the B, and then there's a third option, which is just a small I, which means they're not going to express any of those antigens, and so they won't have A or B antigens. They won't have A or B blood. Right? They'll have type O blood. Okay, and this is just looking at the frequency of the different blood groups. Don't need to know that, but it's just there for interest. And so let's take a look at this one. Okay, so go ahead and work through this one. With the people on your team. We've got about just three minutes left, so I'll probably just leave you in the rooms for probably just about a minute. Just check your answers, and again, you're going to send in this one here that's on the slide. Okay. All right. Let's figure that one out. Easy does it, guys. Got her down? Cool. All right, so you send it in the likelihood here of type O blood. So type O blood would mean that they don't have A or B antigens on their red blood cells. Okay, I think almost everybody's back. A few more people need to send that in on the app there. Go ahead and send that number in if you haven't yet. Again, you're sending it in the percentage format after you've already times it by 100. Okay, I think everybody's back. So I wrote in the Punnett square there. Okay, let's look at the genotypes there. 0, AA, 1 on the AB, 1 on the A with just the small I, nothing on the BB, 1 there, and then 1 for type O blood. Okay, so then we convert that to phenotypes. Okay, so type A would be this one, right? Type B would be this one. Okay, type AB would be this one, and then type O would be this one here. Okay, so that's that example there, and let's see what you guys said on the app here. Good, 98% of you saying 25%, and that's absolutely correct. Almost everybody, great job. Good, okay, and then just lastly, real quick, all this slide really says is I've presented you a simple approach to genetics today, and all of it's true, but it's just it's kind of looking at the ones that are passed down in a simple way. For many things, like even as simple as height or hair color or skin, or definitely something like intelligence or disease risk like cardiovascular disease. For type 2 diabetes and cardiovascular disease, for example, we know of about 50 linked genes that are connected to both of these, okay? So it's not just one, you got the big C or the little c, you got cardiovascular disease, it's not like that, right? It's this huge conglomeration of a bunch of different genetics, and then how those interact with the environment that really give us the idea of whether or not you're gonna have that disease or not. But like I said, there are those things we just looked at that do have that simple Mendelian genetics as the way that they work. So it's a mix of both of those, okay? Okay, guys, so that's it for that today. And then we're going, just one reminder, at the end of I posted the notes or the class on Friday, and I continued on and I went through a little bit slower on that last slide for the TBL activities, which was how to figure out the flow from DNA to the other strand of DNA to RNA and then to the amino acids. Okay, so I went through a little bit slower on that. So if you don't have to watch the whole video of class on Friday, but if you want to zoom to the end there, then you can at least, if you're kind of struggling on how to figure out that information flow stuff, then hop to the end of that video and I've walked through it a little bit slower there. Okay, all right, guys. Well, we will see you on Wednesday if you want to come for class on Wednesday. That's just a review session, but I'll also post a lecture for that day that'll be posted before class as well. Okay. Have a great day, guys, and the rest of your day. And again, if you want to hang out for a sec, if you have questions, please do. See you soon.
Bye. Thank you.
Bye-bye.
Thank you.